today to take you on a sort of a journey that talks about broadly what epigenetics and social epigenetics in particular is, then moving towards showing you some what I hope are really instructive examples that give testament to the fact that indeed, as Jennifer mentioned, early life social experiences can get under the skin to fundamentally alter the way our genes and our biology works, and then hopefully ending up with some interesting discussion about what this means in the bigger picture. So the definition of epigenetics you can see here on this first slide. It basically says that it refers to persistent and heritable alterations in genomic information that do not involve changes in the DNA sequence. And we can talk about this a little bit more as we move forward. But what really the key of today's presentation is that we're talking about social epigenetics, and that is really the process by which life experiences influence the ways our genes are expressed. And perhaps the best way to think about this is that we experience things in our life all the way from birth to before birth sometimes into adulthood. We are focused on the early life experiences, how they influence biological pathways, and how they ultimately then affect the way our genes are expressed. But of course, it's not as exciting as this journey is from the experiences to literally getting under the skin. There is also, of course, the reverse. We're going to talk about both of this, in that perhaps the way our genes work might affect our biological pathways, and by doing so might alter our behavior. So really, the field of epigenetics has become extremely popular over the last 5 to 10 years. While initially it was the domain of people that were interested in model organisms, and now has become really a field that more than perhaps any of the other fields in molecularology that I'm aware of has united and integrated all kinds of disciplines. And so, ranging from people that are interested in model organisms such as rodents, you can see on the bottom left there, or genetic diseases. Those of you who like cats might recognize this calico cat in the middle there. It's a prototypical kind of example of epigenetic biology at work in real life. And today we're going to talk primarily about two things, and that is where epigenetics has become increasingly important, and that is, first of all, the interactions between parents and their environment, and also, as illustrated at this top, on the right top. But also, we're going to start about the influence that the places that we live, and you can see a map of British Columbia on the bottom right there, might have on to our genes through epigenetic mechanisms. So, while I have shown you some definition of epigenetics and social epigenetics, I really, despite being German, and by virtue of being German, was potentially supposed to love definitions, I actually do not like definitions. So for me, epigenetics is something much more fundamental, and that it really is a journey through life. And that is because from the moment we are conceived, you can see on the left here a fertilized egg, during gestation, when we're in our mom's belly, or when we're a cute toddler, such as this cool fellow in the middle, it's my lovely son Sebastian, or perhaps a annoying teenager, which I'm sure none of us ever has been, or ultimately a wiser older gentleman, such as the fellow on the right, Conrad Waddington, who in the 40s and 50s really coined some of the key processes of epigenetics. What we ought to realize is that of the 25,000 genes that we have, their sequence really doesn't change all that much, save for, of course, some rearranging of the immune system, or the occasional mutation that arises during the lifetime. But I think we all agree that across this journey through life, we change quite a bit. And so what happens is that these changes are really rooted in the developmental programming of the epigenome, which really happens in all of us, and occurs from beginning, from fertilization all the way till we eventually die. Now, this is a really exciting field, and numerous people have made terrific contribution to this field, but with all due respect to all of those contributions, it's not the main reason why epigenetics has become so popular. The main reason why it has become popular is that it has become clear that on top of this developmental programming of the epigenome, we can have environmental influences that tweak with the epigenome. And those are going to be the focus of today's presentation. And these environmental influences, as I said, can be pretty broad, ranging from things like the places we live, as we'll see today, as well as the way we interact within our families. And so the way I like to think about the epigenome, Jennifer alluded to the fact that it's potentially very complicated, but I really believe it doesn't have to be. So what I would like for you today to think about throughout this presentation is of the genes as light bulbs. So we have 25,000 genes. As I said, we get one copy from the mom, one copy from the dad at fertilization. And each of those genes, the way I like to think about it is that they can be either totally off, like this light bulb on the right, or totally on, like the light bulb on the left, or somewhere in between, like the light bulb in the middle. And so the question then becomes, what sets the activity of the light bulbs? And if you live in a fancy house, you might have a dimmer switch rather than just an on-off switch. And so it's the switch basically that's shown on the bottom, and this particular dimmer switch would be the epigenome. So in the situation where the light bulb is completely on, the dimmer is totally up. In the opposite situation, the dimmer is totally down, or it could be anywhere in between. So the dimmer, the epigenome, sets the activity of the light bulb, which would be representing the genes. And keep in mind that we have 25,000 of those. So in each of our billions of cells, you can imagine we have 25,000 light bulbs. Now, one of the key mechanisms that sets this dimmer is a chemical modification to the DNA itself. It's a small chemical group consisting of one carbon and a few hydrogens called methyl group, and this can be attached to the DNA itself. And in the following animation, we'll learn how this methylation regulates the dimmer, and by doing so, it basically regulates the activity of the light bulb. And so on the right, you can see a strand of DNA. Now imagine we get a methyl group that comes on to it. You see that on the top there. It pushes the dimmer down. We get another methyl group, pushes the dimmer down further. Of course, the light bulb gets dimmer and dimmer as well. And lastly, we push the dimmer all the way down because we've fully methylated this strand of DNA, and we have turned the light bulb off. So this is what I like for you to think about throughout today's presentation. Think of the dimmer switch as the epigenome, and its ultimate result being the light bulb that represents the activity of the genes. So just to recap, so this we basically have then various states of genes. On the most extreme, we have a silence gene that makes no basically the light bulb is totally off. The dimmer is pushed all the way down because it contains many of these methyl groups. And in the most extreme other end of the extreme spectrum, we have a very active gene where we have no DNA methylation. The dimmer is all the way up, and the light bulb is turned all the way on. So with this, let's continue our journey and think about how this works in the human body. So what you can see here is then basically two different cell types and of the human body. So on the left, you see this is a neuron, a brain cell, and on the right, you see a blood cell. And I hope that you all can see that these cells do look very, very different, despite having, of course, the identical genetic makeup. But as we just learned, what's different in these cells is that of these 25,000 light bulbs, they have very different patterns, and that's shown here. And to make it easy for you, we only show 25 as opposed to 25,000 light bulbs, but you can easily imagine that those would be 25,000 in each of our cells. And so you can see that some light bulbs, like the one at the very left, are similar between the two cells, but others are totally opposite, like the one on the very right. And now this is what happens during development. So we basically, as we develop, we create these patterns of light bulbs in these different cell types. And as I think I mentioned, we have roughly 200 of those different cell types, if not more, in the human body. But now, what happens if, say, we experience a particular environment in early life? What happens is that some of these light bulbs might change. And in the first case, then I'm going to show you what they're going to change in both of those tissues. So you can see now how the light bulb at the very left became yellow in both of the tissues because we experienced, say, stress, or we experienced low socioeconomic status. But there might be also some situations where these environmental effects have different opposite effects depending on the tissue, the cell type we look at. So in this particular case, you could see that on the brain cell, if I take this back and bring it up again, in the brain cell, the bulb became brighter, and in the blood cell, it was turned off. So keep that in mind as we go through this situation. But basically, what I want you to realize is that we do create these different light bulb patterns during development, but then the key thing is that some of those might be changing in response to environments, and those changes might or might not be specific for the different cell types. Now, what does that really mean for us in terms of health and disease? In this next slide, I have a picture of 5 lovely young mice. Those are mice that were just born. And I trust that all of you see that these mice look very different. If there's anybody in the audience that doesn't see that, I might recommend that you see an eye doctor sooner or later. So if you were to see this in humans, if you were to see humans with blonde versus brown hair, like the yellow and the brown coat color of these mice, of course you would think this is genetic, right? They got different genes from their mom or their dad. That's why they have different coat color or hair color, if you wish. What I'm going to tell you is that these 5 mice are actually genetically absolutely identical. They're literally clones of each other. They were bred in the laboratory to be genetically, have no genetic differences. So then the question becomes, well, what really makes those mice different from each other? Why is the mouse on the left having a yellow coat color and the mouse on the right having a brown coat color, and the ones in between sort of mixed and mottled colors? Well, think back to our dimmer and light switch. What happens is that conveniently enough, on the yellow mouse, on the left, one particular light bulb is totally turned on, the dimmer is totally up. Okay? Now watch what happens on the mouth as we go to the right. You can see that the dimmer is pushed down and the light bulb is turned off. So that is fascinating. What's even more fascinating is what actually causes this regulatory change. And it's actually the nutrition of the mom during pregnancy. So depending on the nutrition that the mom gets during pregnancy, we can have the dimmer totally up and the light bulb totally on, or pushing all the way down to basically turn off the system. And there are various nutrients that can do this. Initially, this was work done by Randy Turtle at Duke University. Initially, it was the amount of particular amino acid methionine in the diet, but since it has been shown that as diverse things as fetal alcohol exposure and bisphenol A exposure can also basically affect the balance of the dimmer switch and the light bulb during when the fetus is exposed to it during pregnancy. So what this really means is then that the environment, in this case the environment in utero, regulates the dimmer switch and by doing so regulates the intensity of the light bulb, providing a wonderful example of how this early experience, in this case before birth, actually can literally get under the skin to create different hair color in mice. Now you might ask yourself, what do I really care about hair color in mice? But the key thing is when you then, and you will appreciate it in a second when I show you the next slide, when you let these mice grow up, see what happens. So now we're just looking at the extreme, we're looking at the yellow mouse and at the brown mouse. And now what you can see is this is basically if they were adults, human adults, you can see that the brown mouse stayed fit and healthy versus the yellow mouse became obese. It develops all kinds of health-related, age-related problems. And this, again, in genetically absolutely identical animals. So again, suggesting that this experience, the early life, the prenatal experience, changed the dimmer switch. By doing so, it changed the intensity of the light bulb. But it did so, and that's the key, it did so in a very persistent manner that actually stays with these mice way beyond the original exposure phenotype during pregnancy, all the way into adulthood, to lead to profound differences in health and disease. Now, again, one might wonder that this is rodent research, and it's what's the relevance of this for human health and disease. It turns out that there are numerous examples now that report very similar effects in humans. One of them is shown here. This is data from the Dutch famine at the end of World War II, where women grew up under very low, very dire conditions, where they had almost nothing to eat. Nevertheless, some of them became pregnant, and when researchers looked in their children, who by then were 60 years old, so 6 decades after they were born, after they were exposed to these conditions, what they found is that in their white blood cells there was a particular demo switch that was directly correlated to whether or not their mother starved at the end of World War II. Again, very similar, suggesting that these early life experiences got under the skin to regulate this demo switch in a way that could be seen 60 years later. And interestingly enough, this particular demo switch regulated a light bulb that was is involved in insulin metabolism. Similar, there are a number of data now out showing that in children, one can see a pattern of dimmer switches that can be directly traced back to whether their mother smoked or not during pregnancy. Furthermore, work that we're going to elaborate on, but really seminal work initially from Michael Meany and his group in McGill University, showed that the initially in rodents and later in humans as well, that the quality of the maternal care affects the epigenome of the child. And we'll show some examples from our own work here at HELP in the next little while. Lastly, of course, the closest we can get in humans to these genetically identical mice are monozygotic or identical twins. And many of you might know identical twins. And if you think about them, you might realize that, you probably realize that despite them being very, very similar, they do have some distinct features, including sometimes earlier or later onset of diseases and or different behavioral characteristics. And what work from Petronis in Toronto and Manuel Estella in Barcelona has shown is that while in young identical twins, their epigenomes, so their demo switch patterns is very, very similar. If you look at older identical twin pairs that are 50 or 60 years old, their demo switch pattern actually is quite distinct, and in particular if they happen to have been separated early in life. Again, suggesting that the early life environment got under the skin to persistently alter the demo switch pattern in these monozygotic twins. So collectively then, it's really to the idea that epigenetics is a real integral part of human health and disease. You know that your field has become pretty mainstream if it's featured on the title of Time magazine. So this is from, I think, 2 or 3 years ago, a wonderful article that looks at epigenetics, biology, and from a sort of epidemiological perspective, and the title, somewhat wrong, is it's Why Your DNA Isn't Your Destiny, and talks about the quote-unquote new science of epigenetics. What we like to do in our laboratory and at HELP and in the framework of partnerships that we have is to really think of epigenetics as an integrative model affecting health, disease, and behavior together, of course, with the environment, and that's going to be the focus of much of the rest of today's presentation. But we're also cognizant that, of course, our genetics, our genetic makeup is both linked certainly to the way we interact and respond to certain environments, but more recently, we and others have also found that there is actually quite an interesting linkage between the genetics and the epigenetics. Okay, so we then basically, having set out this paradigm, we started to ask in populations from the lower mainland initially, what are some of the characteristics of the epigenome, focusing again on DNA methylation, the dimmer switches that we talked about earlier. And in work that has been published recently, we basically first ask, are there any demographic or other variables in white blood cells of adults from the Vancouver area that we can link to their dimmer switch pattern? And indeed, as you can see on this next slide, we found numerous of those. So what you can see here in these graphs, I want to briefly explain, those are p-value distributions, so likelihood distributions. Anytime you see these bars skewed to the left as opposed to that flat dashed line, it means that there is a correlation between a given variable and the settings of these dimmer switches. And so in all of these cases, you can see that the very left and the second to the left column are much higher than the ones on the right, so indicating a strong skewing of the likelihood distributions, and thus suggesting there's a correlation of things such as gender and ethnicity with the dimmer switches. This was perhaps not surprising because others have found this as well. But more interestingly, for the purposes of our own interest and work, was that we also found that things such as perceived stress, so whether you reported to our research team that you were stressed or not, or more molecular level output of stress such as total cortisol, so in the lower left corner graph here, all are correlated with the setting of dimmer switches in adults from the lower Vancouver mainland. And so that, especially the last two, were really interesting to us because they suggested that psychosocial factors can be associated with dimmer switch setting in white blood cells in the cells that are relevant for the immune system in living humans. And it really fit very well with a research field that we had developed in parallel to these investigations to, as Jennifer mentioned, to understand how early life experiences get under the skin. And we have done, our research team had the privilege of really standing on the shoulders of giants here to do this and develop this research program. So we have been working very closely with folks like Greg Miller and Edith Chen, who used to be in psychology here at UBC and now at Northwestern University, Tom Boyce here at the Human Early Learning Partnership, now at the University of California, San Francisco, and Maryland, Essex at the University of Wisconsin. But perhaps most importantly, we have been had the privilege of working with the late Clyde Hertzman, who passed away untimely and way too early about a year ago, the former director of the Human Early Learning Partnership. And Clyde was a true mentor, a spirit, a friend, and really one of, if not the worldwide leader in understanding how this genomic embedding happened. So collectively, we're really trying to understand then how these early life experiences, such as early life parental stress and early life socioeconomic status, get under the skin to perhaps affect childhood temperament, behavior, and trajectories, most importantly. I really like to dedicate then the rest of this lecture to Clyde's legacy because his thoughts and his insight and his leadership were really instrumental in developing these concepts. Now, what we first asked is the early life socioeconomic status. And the reason for that is that shown in this graph. This is data published some years ago by others. What you can see here are basically the probability of coronary heart disease among more than 1,000 white male medical school graduates from Johns Hopkins University in the United States. And for those of you who are at all familiar with the United States medical system, you will realize that these people are extremely well educated. They're most likely in very high socioeconomic status groups and are very well respected in their communities. But what this graph actually shows is two groups, and you can see that the upper group has a much higher probability of having coronary heart disease than the lower group. What distinguishes these groups is really only, this is after correcting for all other factors, is really only their socioeconomic status in early childhood, in the first 5 years of their life. With the folks who grew up in low childhood SES having a up to 2.5-fold higher risk of developing coronary heart disease than the folks shown in the lower graph that grew up in high childhood SES. So really suggesting in a very real example that the early life socioeconomic status does get under the skin. It matters for lifelong health, whether you grow up like these folks on the left picture or on the right picture. And then the question, of course, becomes how. And if you followed me at all so far, you will realize that, of course, our hypothesis was that part of this mechanism might be setting the dimmer switches and changing the light bulbs in our body. And we investigated this directly in work that has been published 3 years ago with Craig Miller and Edith Chen, where we showed that basically in a group of more than 100 people from the lower mainland, we find a biological residue of the low early life social class. And in particular, the way this study was set up is that we had basically two groups of adults that were distinguished by their childhood socioeconomic status, in that half of the people were of low socioeconomic status and the other half were of high socioeconomic status. Their socioeconomic status in adulthood, as you can see in this scheme on the left, was mixed, so that we could directly ascertain the effects of the childhood socioeconomic status. And in this particular study, we initially focused on the light bulbs. And what we found is if you compared the 25,000 light bulbs in the white blood cells of these people, that there were indeed differences between the two groups. These differences weren't just random. For the experts among you, they were basically indicative of a heightened immune response in these people that grew up in low early life SES, based on bioinformatic analysis that suggested that certain transcription factors, such as this thing called GR, glucocorticoid receptor, or this NF-kappa B, which you can see on the left, on the y-axis there, were differentially regulated in these people. Now, we're in a very good situation because we actually had also collected immune cells in parallel to measuring the light bulb activity from those people. And we could then challenge these immune cells to directly test whether indeed the immune cells of the folks that grew up in low early life SES were more reactive to challenges such as that things that, for example, bacteria would impose onto our immune system. And as you can see on the right upper two graphs, indeed, there was a statistically highly significant difference in the way these immune cells were basically reacting to these challenges. In that, again, as predicted from the light bulb pattern, the folks with low early life SES had a heightened immune response when their immune cells were exposed to these challenges. Similarly, and consistent with it, as you can see in the lower right graph, the folks that grew up in low early life SES also had a heightened stress response as judged by the cortisol output throughout the day compared to the folks who grew up in high early life socioeconomic conditions. And lastly, the question, of course, then becomes how is this linked to the dimmer switches? And you can see this in this next slide here. Very clearly, the early life SES has a highly skewed p-value distribution, as you can see to the on the left graph here. So suggesting that indeed these early life socioeconomic conditions also change the way the dimmer switches are set. Interestingly, we did not find any correlation as judged by the very flat p-value distribution on the right between current socioeconomic status and these dimmer switches, suggesting that it really matters more what kind of socioeconomic status you grew up compared to what socioeconomic status you have in adulthood. So collectively, what this really means is if you grew up in early life SES, you had people have a distinct dimmer switch setting that is linked to their low early life SES or high early life SES. And in particular, these dimmer switches then likely regulate the light bulbs in such a way that they give rise to a heightened inflammatory gene activity pattern, which can be seen not only on the level of the light bulbs, but also on the level of the actual functional response of the immune system, and lastly on the level of the stress response in these adults. And again, this is all irrespective of current SES. And I think this is a beautiful example of multi-layered example going from the dimmer switches to the light bulbs to the function of the immune system to really suggest that the early life poverty gets under the skin. Now, of course, I don't want to leave you here with a depressing message that low early life really is all that detrimental without giving also perhaps some idea of for hope. And this is shown in the next slide. So what we found in focusing on this group of 50 low early life SES people and asking a simple question, is there anything that can provide perhaps hope that can provide perhaps guidance for interventions. And what we found is that it was actually the level of the maternal warmth, and in particular, if these folks had received high levels of maternal warmth, they became much more resilient to the effect of the detrimental effects of the low early life SES. And that can be seen again on the level of the to some extent on the level of the light bulbs, but perhaps most importantly on the level of the immune reactivity as shown in these two upper graphs, where you can see that in people of low early life SCS, that also there's a stratification in terms of the immune response, and that people that also reported low levels of maternal warmth still had the high immune response, but people who reported high levels of maternal warmth had much lower immune response, consistent with the fact that the maternal warmth would buffer up in part these detrimental effects and provide resilience. So basically, what I always advise, if you happen to grow up in dire socioeconomic circumstances, if you receive a lot of maternal warmth, at least on the level of this genomic embedding, you might be okay. And so I do recommend to choose your parents wisely. Now, of course, as exciting as this research was to us and to many others, the one limitation certainly was that we interrogated adults asking about their early life upbringing. And even though the parents in this particular studies were also interviewed, it's not that easy to reconstruct what happened 30 or 40 years earlier in terms, especially not in these early years. And being a fairly new parent myself, I'm sure that no parent would admit they didn't love their children much. And so this really was one of the limitations that we discussed when this paper was published. But as I always tell my students, good things happen when you talk to people. And so one of the good things that happened is that through Clyde and Tom Boyce, we met Marilyn Essex, and Marilyn really has made a living through a wonderful study called the Wisconsin Study of Families at Work. And that's a longitudinal birth cohort, meaning that Marilyn has followed more than 500 families in Wisconsin from before birth all the way into late adolescence. These children are now 16 to 18 years old, and has really collected along this journey just a fantastic amount of really high quality data that were objectively ascertained by her research team. And luckily for us, Marilyn also had collected DNA at age 16, which then allowed us to ask, in this very well controlled circumstance, well, was there anything in this group in the early life that also was correlated with their demoswitch setting in late adolescence? And of course, the first question we asked, based on the last few slides that I just showed you, was socioeconomic status. The problem with this study, quote unquote, the problem with the study was that there is very low levels of socioeconomic status diversity in the cohort. Basically, it's a lot of middle-class family from suburban Wisconsin places, and so we did not find anything because there was really no gradient in socioeconomic status in this cohort. But what we found is that there was actually quite a gradient of parental stress. And so we asked, in these demoswitches in these adolescents, is there anything that is correlated to the level of stress that their parents had during their early years? Very consistent with this idea that I put forward in the last slide, that the level of maternal warmth was somewhat correlated with the light bulbs and potentially the demoswitch settings. And indeed, what we found is that some really interesting patterns in that when we ask in this demoswitch setting in adolescence, when we look for correlations with the parental stress, we found, first of all, that there was a very good correlation between the mom's stress and these demoswitch patterns during infancy. So you can see that 139 demoswitches were correlated with the level of the mom's stress. The HCS stands for high confidence sites. This is simply a statistical assessment of how confident we are about these measures. The others you will see, MCS stands for medium confidence sites based on statistical ascertainment. We found very few, if any, remnants of paternal stress during infancy on these in these adolescents' demo switch pattern. However, when we looked a little later during preschool, we saw a dramatically different picture. What we found is now that there was basically no correlation, at least in this cohort of 110 adolescents, between the level of the maternal stress and the demo switch settings. But instead, what we found is a correlation now with dad stress. So for those dads of you who hope you would get off the hook, you don't. There is actually a so the data then suggests that even in the full group, when we basically we look at both boys and girls, we see some correlations, but it becomes much, much stronger. And you look at the numbers here, especially on the medium confidence sites, when we only look at the girls, suggesting that in preschool, the dad's stress might leave an imprint on the demo switch setting, in particular in girls. So really, what this data, this somewhat complicated slide says is that there is, we can see a vestige of early parental live parental stress on demo switch setting in adolescence, and this is not random. It's very specific for the developmental stage. In infancy, the maternal stress matters, and it doesn't matter whether it's boys or girls. And in preschool, it's the dad stress that matters, and it matters primarily actually for girls. And that's very consistent with what experts will tell you is known in the field of child development as it relates to the interaction between children and their respective dad or mom in these early years. So this work is published, and I'm happy for you to send you the paper if you want to learn more about it. But just one example then, for two particular demo switches that regulate the same light bulb. You can see here on the y-axis, this is the DNA methylation, so the setting of the demo switch, and on the x-axis is the maternal stress at infancy, and you can see the regression line basically is giving you some idea of the strength of the correlation that we have here. Now, of course, we're very excited about this because basically what it says is it's yet another piece in the puzzle that suggests that indeed the early life environment gets under the skin, in this particular case the parental stress level, to affect these demo switch settings. But of course, perhaps the even more exciting question then is, well, is this in any way, shape, or form actually associated with children's behavior? And because of the depth of analysis that Marilyn Essex has done in this cohort, we can then really move on to a child-centric analysis of these data and ask whether any of these demo switches are related to whether a child is shy and fearful and retrieved like the boy on the top, or open and outgoing like the girl on the bottom, either in preschool, kindergarten, and grade 1. Now, this work has not yet been published. We're currently writing it up for publication, but I'm of course only putting it on there because we did indeed find these kind of relationships. I want to show you just one example, and that relates to grade 1. In that we found that there was a very strong correlation in that more than 246 demo switches were correlated with the observed temperament of the child. In this particular case, it was anger. And we're most excited about this data because for many different reasons, but one of them is that we found several demo switches that regulate the same light bulb, which to behave the same way, which always gives us extra confidence. But we also found that some of these demo switches also were correlated with anger already when we look back at preschool, suggesting that perhaps there is indeed a persistent pattern of these demo switches that is related to children's behavior. So stay tuned for this. Hopefully next time we have a webinar, this work has been published and I can tell you more about it. So just to summarize then, what I hope I have shown you today is that certainly epigenetics has become an integral component of human health and disease. And we touched upon some of the key principles, but more importantly, perhaps, I hope that you're equally excited as we are here at HELP about the potential for epigenetics to explain the genomic embedding of early life experiences, and in particular, things like the biological residue of early life poverty that we talked about. We talked about these demo switches being associated with the early life parental stress. and lastly touched upon the fact that perhaps indeed they're also correlated with childhood temperament and behavior. So then to just summarize this in another way, perhaps the way we like to now look at this is that we like to look at this relationship between the dimmer switches and the light bulbs as being central to mediating early life experiences, whether it's early life parental stress, early life socioeconomic status, or perhaps even prenatal experiences with behavior and phenotypes in children and potentially in adults. Of course, we are cognizant that this really needs to be looked at in a developmental context, and as we briefly touched upon, also in the being cognizant that there are different tissues that might have different responses to some of these environments. And lastly, as I touched upon, we are certainly very interested in understanding the interface between the genome, the epigenome, and the environment. So then to basically leave with you perhaps somewhat provocative question, what does all of this mean in the context of nature versus nurture? Is this idea really supported by this data, or is it perhaps much more that these things interact with each other? And so I'm happy to discuss our views on this. And lastly, I want to just acknowledge the work, the people who have done all the work. I think I touched upon all of our collaborators shown in the gray box. But of course, I'm only sitting here and talking. It's really the people, the wonderful people in my laboratory that you see on the top here that have done all this work. And at the bottom you see all the agencies that have generously provided funds for us to do this work. So thanks a lot for your attention, and I really look forward to some fun and discussion. Thank you.
Okay, great. Thank you so much, Dr. Kobor. That was excellent, as usual. Mike has presented at a few of our conferences in the last few months and done a great job. Every time it improves. So we'd just like to hear from you. If you have any questions, just type them in on your screen, and I will be moderating these questions over to Mike. One of the main questions that we usually get is this presentation going to be available after today? And we have recorded the audio, and along with Mike's slides, that will be available on our website probably early next week when I get a chance to upload it to the website. So if you just go to our website, earlylearning.ubc.ca, and then you click on the Events tab, and then you'll go into Past Events, and just look for Mike's face, and right next to his face, you'll see the title of the webinar, and you can click on that, and it will go straight to this video. So if you missed anything, or you want to hear the presentation all over again, or share it with friends or colleagues, you can go ahead and do that next week. Okay, we have a few questions coming in here. Are governmental powers aware of these data, and are they responding appropriately with changes to funding for early childhood care services and supports for families to reduce stress? For example, early kindergarten entry, universal child care, and Paul Kershaw's work.
So that's an excellent question, and it's something that we're really starting to think about very, very hard. It's certainly something that was dear to Clyde's heart, no doubt about it. I want to perhaps just caution a little bit that what I've shown you today is really pretty new research. And we are really in a wonderful position here at Human Early Learning Partnership at UBC that we're one of the few places that really has deeply investigated those things. But even so, there is still so much more to be learned, and we certainly need to be aware that we want to reproduce these findings in different populations with different methodologies. But ultimately, just like you expressed, I very much hopeful, and it's certainly part of what we're trying to do at HELP, to bring this kind of research into the public and into the policymakers, attention, to perhaps provide yet another quote-unquote molecular evidence for the importance of really providing the best possible childhood experiences to our children that we can.
Excellent. Thank you, Dr. Kobor. And just to add to that, as you will appreciate, this is very cutting-edge research, and so there hasn't been a lot done on the side of translating this knowledge to communities and to policymakers. So the knowledge translation team here at HELP, we are working on uploading more content to our website and designing a few research briefs that will act as one-page handouts that a lot of you can use to translate this knowledge to the people that you work with and to families. So watch for that. That should be coming within the next few months, and we'll happily send you links to that when we have it.
And perhaps just to make a very brief addition, I'm more than interested in feedback and advice and ideas that you might have on how this kind of research is best communicated and shared with both the public but also policymakers. So feel free to also be in touch with me, along with the wonderful team of KT at HELP.
Mm-hmm. Okay, next question. You talked about the impact of prenatal nutrition on a child. What does the research show about the impact of prenatal stress on a child?
Yes, again, that's an excellent question, and I am very confident that in the next webinar I will be able to answer this to you, this question better, because it's one of the key questions that we're currently investigating, both in the Wisconsin cohort that I touched upon, but also in a wonderful cohort that we're involved in, the Canadian the child study that in part is taking place in the Lower Mainland, but also in 3 other sites of Canada, where we have the opportunity to look exactly at these questions and ask them and answer them hopefully in the most rigorous fashion. My prediction would be that perhaps the effects might be even stronger than the ones we see for early life stress.
Okay, great. So just to follow that up as well, there are a list of peer-reviewed journal articles on our website. If you go to Dr. Kobor's page, you'll see all of the publications that he has written, as well as Clyde Hertzman's work is on our page, and Tom Boyce. If you're looking to search just for epigenetics work, you can go to our publications tab and just search the word epigenetics. If there's anything on there that you don't see or that Mike mentioned in the presentation, feel free to email us at any time and we can send those articles off to you. Any more questions? No, I don't see any more questions coming in. Last chance. Oh, here we go. In regard to the Wisconsin study, in your opinion, does the research related to the effect of father stress on preschool children's health make a case for policy change in supporting early years father support funding programs?
It's an interesting question, and again, I would certainly the fact that the fathers seem to matter more in terms of the effect on demonstrative setting would argue in favor of investigating this further and perhaps transferring it to public policy. I actually recently got a really interesting question that is very related to this one, and that is if we look at the effects, the correlations that we found between the mother and the children's demonstrative setting in infancy. Somebody asked me whether this could actually be the dad if it's purely a reflection of that the mom generally is at home during infancy as opposed to the dad, and how that would be, whether it would actually be the dad who would be correlated if it was in families where we had a stay-at-home dad. So that's a related question that I think there's some interesting natural experiments that could be done given the changing times. Keep in mind that the Wisconsin study was done 18 years ago. In terms of
so somebody had mentioned Paul Kershaw's work. Dr. Paul Kershaw works at the Human Early Learning Partnership, and his work is much more centered on policy. We're a very interdisciplinary team here at HELP, and so Mike's is obviously related a lot to biology and epigenetics, and where the policy work comes in is Paul's expertise. So if you're looking for that kind of thing, he writes a lot about parental leave time and more focus on fathers staying at home longer with their children. So you can find that information on our website, or you can go to gensqueeze.ca, G-E-N, squeeze.ca, and you can find their policy recommendations, which are very much in line with the research that Dr. Kobor has done. Okay, one more question. Is there any particular research about the biological embedding of violence that you can recommend?
So there is work from Michael Meany, I think I mentioned Michael's name, and Gustavo Turecki from McGill University that was published some years ago that basically is the closest that I can think of at the top of my head of that provides some evidence about violence, and what they have shown in a very, very small cohort of, I think, 10 suicide completers is that there was a epigenetic pattern in the brain of these suicide completers that could be traced back to them being abused early in life. Now, I don't remember whether, I think it was physical abuse, but I don't remember exactly the details, but that's probably the most provocative piece of published evidence that I can think of that would argue that there is some imprint of early life violence. We in our lab and at HELP, we're involved in a number of collaborative studies, including some that look at kids that were adopted from very abusive and dire orphanages in Eastern Europe, and so that have now relocated to the United States. So not exactly the same circumstance, but we're certainly trying to get to some of these questions as well.
Are there specific economic income cutoff points that link accurately to the negative or the positive epigenetic outcomes that you mentioned?
That's a very good question. It's always, so socioeconomic status as defined in our studies, and this is done by psychologists who are experts in this. It's using a British scale for socioeconomic status, which isn't purely based on income, but it's also involving things like education and whether you rent or own a home and things like that. So it's a bit more complicated measure than just purely measuring the amount, reporting on the amount of dollar that a family earns. I should say that based on these studies that I showed you, we have now received funding from the National Institute of Health in the United States to replicate this study in a much larger group, and which with more granular measures, including more granular measures for socioeconomic status. So that's very much ongoing, and hopefully it will be finished in the next year or two.
Excellent. And then just a couple of comments. Universal early childhood education will probably be the solution, since you say the maternal warmth is very important, or how to interact with a child, especially in low SES situations. And somebody else wrote in, if anyone is interested in hearing Dr. Kershaw speak, he was on CBC radio this morning in Toronto and spoke very eloquently about the Generation Squeeze campaign. His interview is on the CBC website. Is there any research that you may recommend reading about epigenetics and suicide in young women?
Again, I think to my knowledge, and the only work that I'm aware of about suicide is the work from Michael Meany and Gustavo Turecki as it relates to epigenetics. That's not to say there isn't other work out there that I might have missed, but I don't think their work specifically distinguish between females and males because the group was so small to start with. But it's certainly something that is very interesting. Overall, for this field is to understand whether there are differences between male and females. And I think I touched upon some of in terms of the epigenetic residues of the parental stress. I wouldn't be surprised if there are differences in terms of behavior, including extreme behavior such as suicide, that might be different between males and females and might be linked to some epigenetic differences.
Okay, great. Doesn't look like there's any more questions here. So just to remind everybody, this is a very new field of science, and we will be trying to promote more and more of this research as it keeps coming in from Dr. Kobor's lab, and hopefully be doing more webinars in the future and presentations into communities or at conferences. So just keep an eye on the health website, and you can find this presentation once again there for viewing on our past events page. Thanks so much, everybody, for joining us, and hope you can join us again soon.
I also want to say thank you for spending an hour with us, and hope you enjoyed it. And I certainly did enjoy the questions, excellent questions all around. And hopefully we'll be in touch, and feel free to engage with us as much as possible. So thanks a lot, and have a nice day.